All right, everybody, welcome to this week's video lecture. Today we're going to be covering the peripheral nervous system. And with that, let's go ahead and get started. So I am breaking this lecture into 4 different sections. And so with that, I'm going to do 4 different videos just to make it seem a little bit less, I think, overwhelming. And so in this first section, we're going to talk about in general what the peripheral nervous system entails, also describe the general structure of a nerve, define ganglion, and talk about different ganglia in the body, and then finally finish this video out with talking about nerve regeneration. All right, so again, in this section, we're going to start with an overview of the peripheral nervous system. So let's get started. So the peripheral nervous system is made up of all of the nervous tissue that's outside of the central nervous system. And this includes our cranial nerves, our spinal nerves, our nerve plexes, and then also peripheral nerves, which are the ones that we have named. And so if I say the sciatic nerve, for example, that's an example of a peripheral nerve. And the peripheral nervous system as a whole is divided into divisions. So we have the sensory division, which is the afferent division. So meaning it's going to take stuff to the central nervous system. And then the motor division is our efferent division. So it's going to take things away from the central nervous system and deliver signals to the body. So here is a schematic of the breakdown of the peripheral nervous system and its sensory and motor divisions. So notice over here in the sensory division, we have our somatic sensory, which is going to be general touch, pain, pressure, vibration, temperature, proprioception, and all of this is going to be in regards to the skin, the body wall, and the limbs. And the special senses that are associated with somatic sensory are hearing, equilibrium, and vision. And then our visceral sensory is going to do a little bit different things. So this is going to be stretch, pain, temperature, chemical changes, and irritation in the viscera. So this would mean things like our cardiac muscle, our smooth muscle, our glands, our organs. That's all going to be visceral sensory. And the special senses associated with visceral sensory then are taste and smell. Moving over to the motor division of the peripheral nervous system, this also breaks down into somatic motor. Somatic motor is going to innervate our skeletal muscle. And then sort of like we had visceral sensory over here, the other division of motor is going to be autonomic nervous system. And this is going to innervate the smooth muscle, cardiac muscle, and glands to perform actions. And then our autonomic nervous system can actually further be broken down into the sympathetic and the parasympathetic division. And so this concept of the autonomic nervous system as a whole is something that we're going to talk about in next week's lecture. So you'll learn a lot more about the autonomic nervous system. So let's first talk about what a nerve is. A nerve is a cord-like organ of the peripheral nervous system that is composed of bundles of myelinated and non-myelinated peripheral axons, which are also enclosed by connective tissue. And we have 2 main types of nerves depending on where those nerves are originating. So we have cranial nerves and we also have spinal nerves. And we're going to talk in more detail about each of these things. But let's first focus on the coverings that are associated with nerves. So if you remember back to the muscle tissue lecture, we have our myofibrils that are wrapped up together into sort of these functional units, which are then wrapped up into other bundles, which then created our whole muscle. And the nervous system is really similar to that. So we have some connective tissue coverings here, including our ingenium and The endoneurium is the loose connective tissue that is going to enclose individual axons and their myelin sheaths. Okay, so we can see then here the splitting of the endoneurium for one axon and its myelin sheath. Okay, so moving on then to the next sort of protective structure here, we have multiple axons that are going to be bundled together into what we call fascicles. All right, and so the fascicles then are going to be wrapped together via this perineurium. So the perineurium then wraps all of the fibers together into these fascicles. The third sort of structural organization here that we're going to then talk about is the idea that there are multiple fascicles bundled into one whole nerve. Okay, and so this biggest structure down here is going to be the entire nerve, and this one is wrapped by the epineurium, and the epineurium then is our tough fibrous sheath that bundles all of the fascicles to then form this whole structure, and this whole structure is nerve. Okay. So now let's talk about this sensory, motor, or both sort of situation here. So as I previously mentioned, our sensory fibers, these are going to be the ones that are carrying impulses towards the CNS. So everything sensory is going to go towards the CNS. Okay, so this is all the sensory stuff is going towards the CNS, and these are afferent. All right, and then once the central nervous system, we know, processes that information, then the central nervous system will send a signal out, and that outwards signal is called the efferent signal, and that's going to travel along the motor nerves. So we have sensory nerves and we have motor nerves. But we also have these things called mixed nerves, and mixed nerves contain both motor and sensory fibers. And so mixed nerves carry impulses to and from the central nervous system. And just so you guys know, most nerves in our body are actually mixed nerves. So they have fibers that are both sensory and motor. It makes sense to do it this way by just bundling all of those fibers into one whole nerve, so you can transmit sort of like a highway where you have sensory signals going one direction, and then you have motor fibers going the other direction, and they're passing by each other, but they're like still in the same location, if that makes sense. So that is one of the most I guess convenient things about nerves as a whole, is many of them are mixed. And additionally, mixed nerves also carry both somatic and autonomic fibers. So not only are they carrying fibers that are full of sensory information and then sending out motor information, they also have specific fibers that are designated to skeletal muscle sensory and skeletal muscle motor, or like skin sensory and skin motor, and also like this visceral sensory. So organ sensory information gets sent up maybe just alongside of skin sensory information. Okay, so just know that most nerves then, they actually are carrying all four of these fibers. So they have somatic sensory, visceral sensory, somatic motor, and autonomic motor. Okay, so, and just to kind of clarify that here, this visceral efferent is the same thing as ANS fibers. Okay. All right, so the next thing that we're going to talk about are ganglia. And in the lecture last week on the spinal cord, we did see the word ganglia. And so let's talk about what these ganglia are. So ganglia are just structures that are in the peripheral nervous system that contain the cell bodies of PNS nerves. Okay, and so ganglia that are associated with afferent neurons are going to contain the cell bodies of those sensory neurons, of those afferent neurons. An example here, I just told you last week, we saw this in the spinal cord lecture. The example here is the dorsal root ganglion here. That you see on the dorsal aspect of the spinal cord here. You can see this right here is considered the ganglion. It's this sort of outward protruding area. And this is actually going to contain the cell body for this sensory neuron. And guys, remember cell bodies, this is where the nucleus is located. This is where all of our functional components of a cell, in a sense of like proteins, DNA, all that stuff is going to be located up here in the cell body. Okay, so all the really useful stuff. And kind of going on from here, so afferent neuron ganglion have the cell bodies of these sensory neurons, but ganglia that are associated with efferent nerve fibers contain only autonomic motor neurons. Okay, so for example, and we'll talk about this a little bit more next week, but we have autonomic ganglia, which are going to hold the cell bodies of motor nerves that go to visceral locations. So like to our organs, our smooth muscle and our cardiac muscle. And we can see here, this would be the sympathetic trunk ganglion, and they actually line just the side of the vertebral bodies, and they go all the way down. And actually, the autonomic ganglia are of the sympathetic chain are only located in the thoracic region of the spinal column. Okay, so this is actually only present in the around the thoracic vertebra. Okay. And hopefully this makes sense, because if you think about the body as a whole, if we're okay, so if this is cervical, thoracic, lumbar, sacral, if you think about everything that is located right here in this thoracic region, this is where many of our vital organs are. And we know then that visceral things are going to be innervating our organs. So that's kind of just how I associate the, I guess, ganglia here. And in addition, if you also think about our different plexes, notice and remember we had a cervical plexus, and we had a brachial plexus, and a lumbar and a sacral plexus. But there's a really big area of the thoracic region of the spinal cord that has no plexus associated with it. And the reason for that is because instead we have these sympathetic trunk ganglion. All right, so the last topic here in part 1 is nerve regeneration. And this is a really hot topic. It's really good information to know. So in general, mature neurons are amitotic. And amitotic, you guys are not exactly 100% sure what that means. Amitotic means no cell division. They don't divide once they are adults. So if the soma, or if the body rather, of the damaged nerve remains intact, then if we're talking about peripheral nervous system, the peripheral axon may be able to regenerate. However, nerve regeneration, absolutely 100%, unfortunately, does not occur. It does not occur in the central nervous system. Okay, nerve regeneration does not happen in the central nervous system. So let's talk a little bit more about this. And we're going to talk about why nerve regeneration doesn't happen in the central nervous system. So most CNS fibers, they never regenerate. And the reason for this is actually because oligodendrocytes, and if you guys remember what oligodendrocytes do, these are the myelin-making cells of the central nervous system. These oligodendrocytes actually have a protein that is the growth inhibiting protein. And so because of that, these cells cannot regenerate the myelin once the sheath has been destroyed. Now, in addition to having this growth inhibiting protein on oligodendrocytes, CNS fibers, if there is an injury, the astrocytes are actually going to form scar tissue. Okay, so this is going to prevent then the regeneration of nerves in the central nervous system. Now, some potential ways that we can get around it as healthcare professionals. One potential way to do this is to neutralize those growth inhibitors. So basically, what I mean here is we're going to just block these growth inhibiting proteins, and so that we can potentially allow these oligodendrocytes to make their repairs. The other thing here, and this is kind of associated with the previous point, is blocking the receptors for inhibitory proteins. And so this isn't really necessarily a problem on the oligodendrocyte itself. It would be a problem, it would be something that you would be targeting the actual nerve, because the nerve would have the receptors for the inhibitory proteins that are released by the oligodendrocytes. And then finally, the other way to potentially treat CNS nerve damage is to destroy the scar tissue components. So remember that astrocytes, if there's an injury, they form the scar tissue. So finding a way to destroy that scar tissue is definitely going to, I think, improve the outcome. Now, I say treatment here, but realistically, in terms of clinical medicine at this point, we're not necessarily here yet with really any of these things, though I know there's a lot of research going into this. Just know that these are just ideas of how we could maybe regenerate CNS nerves. So even though we can't regenerate nerves in the central nervous system, it is possible to regenerate nerves in the peripheral nervous system, but only if damage is not severe. And so in terms of peripheral nervous system regeneration, the following steps are taken. So the axon is going to fragment, which you can see here's the axon. So this would be closer to the cell body, this side, and then this side is closer to the terminal. And so if there's damage, say here, then what's going to happen is that part of the axon is going to start fragmenting, and then the myelin sheaths that are distal to the injury begin to degenerate. All right, so that's what you see happening down here, is that these fragments of myelin sheath, these droplets rather, this is called Wallerian degeneration. And so this degeneration is actually going to start spreading all the way down the axon. So that's step one. Step two is macrophages, which are just basically little immune cells that are going to eat up stuff we don't want anymore. So the macrophages, you see them in here, they're going to clean out the dead axon debris. And then the Schwann cells, which if you guys remember what Schwann cells are, these are the myelin makers of the peripheral nervous system. These are going to be stimulated to start dividing, right? So they're undergoing mitosis and making duplicates of themselves in order to start creating this new myelin sheath. The third thing that's going to happen then here is the axon filaments are going to start to grow through this regeneration tube. So as these Schwann cells are dividing, they form a tube, and the axon, all of the new fibers, the new little filaments, are going to start to grow into and through that tube. And then fourth and finally, what happens is the axon is going to be regenerated and a new myelin sheath is going to be formed by the new Schwann cells that have just divided. So here's just a big picture of all the 4 steps that I just talked about. And that summarizes section 1. So I will be back for section 2 very shortly. In section 2, we're going to talk about the cranial nerves. And so stay tuned for that.